Good afternoon, everyone. Am I audible? So my voice is reaching to you? So good afternoon, good afternoon. So you had your lunch? Obviously, most of the students are in sleeping mode because having a lot more lunch, or we can say it's a heavy lunch. Okay, no. Let's say in the last class, what are the things we have discussed? So I'm just going for the important points. So one important point is, I'm all good. Maheshwari, how are you? Achha, today I think it's your day of result. Class 10th, right? Good afternoon. Yeah, for state. Okay. So anyway, we'll recap the important points. So 575, 560, oh, good. So we'll recap a few points that, what are the points? Firstly, it's a plasmid. So can we say it's a plasmid? What is the uses of plasmid? And do you have plasmid? Any one of you have plasmid? So obviously my answer is no, because eukaryote is not having any plasmid. And why eukaryote is not having any plasmid? Why eukaryote is not having any plasmid? Just answer me, how many chromosomes do you have? How many chromosomes do you have? So I think answer is 46 chromosome. It's a 46 chromosome. Okay. So everyone is having 46 chromosome, or we can say 23 pair of chromosome. So 46 chromosome means you are having a lot more space to store all the information that is related to the human beings. Related to the human beings, we can store all the information properly, perfectly in 46 chromosome. In 46 chromosome, or we can say it's a 23 pairs. But in the case of prokaryotes, or we can say in the case of bacteria, it is having only one chromosome, or we can say only one double-stranded circular DNA is there. Only one double-stranded circular DNA is there. And that space is not sufficient. That space is not sufficient for what? To store all the information that is related to the bacteria. That is related to the bacteria. So whatever the information left, the bacteria will put into what? Bacteria will put into the plasmid. Okay, so that's why plasmid is also giving what? Plasmid is giving extra, or we can say extra features, or we can say it's a unique phenotypes. So one chromosome is looking different from other chromosome because of having different features, or we can say special features present in the plasmid. Next one is it's a main genetic material that is nothing but DNA, and that too is the double-stranded circular DNA. After that, we were also talking about what? Mesosomes. Mitochondria is not present. Mitochondria is not present in the bacteria. So instead of mitochondria, what is there? So instead of mitochondria, mesosomes are there. So mesosomes is somehow performing the similar function as it is performed by mitochondria in my body, your body, plant's body, or we can say any higher organism's body. So that is a mitochondria, and mesosome is clear. After that, one more is coming, that is the chromatophores. So chromatophore is present in the bacteria wherever it is performing photosynthesis. performing photosynthesis. So obviously photosynthesis requires what? Pigment system. And pigment system is related to what? So pigment system is related to the chromatophore. Chromatophore. So it will simply, we can say fix, or we can say it will absorb the sunlight, and that sunlight is useful for synthesis of the compounds. Synthesis of compounds. Okay. So these are the few parts. Next, fimbriae is there. So fimbriae is, it helps in attachment to the surface. So that is a fimbriae. After that, one more is there, that is known as pili. So pili is mainly for the conjugation, or we can say it's a reproduction, or in other word, we can say the exchange of genetic material between the bacteria, or we can say among the bacteria. So after that, flagella is done. Flagella is meant for the locomotion, or we can say it's a locomotory organ. So these are the few parts that we have discussed in the last class. After that, I think we will discuss some more points that is related to what? Bacteria. So let's talk some points that is related to the bacteria. So what's that? Can you guess? So next one is ribosomes, or yes, polyribosome. Rajgopal, that's correct. Polyribosomes, many other points are there, but polyribosome is also left. So if you are talking about the polyribosomes, so what's that polyribosomes? Polyribosome is nothing but a complex, or we can say a structure. So that structure consists of what? One mRNA. Consists of what? That is mRNA. And mRNA is nothing but it's a messenger RNA, or we can say it's a nucleic acid. And over this messenger RNA, what is there? So over this messenger RNA, ribosomes are present. And ribosome, you all know, having two units. So one is a smaller unit, and other one is larger subunits. So smaller subunits and larger subunit. Likewise, more than one ribosomes are present on the single mRNA. This structure we can call it as a polyribosome. And obviously these two structures are there. So it will help in synthesis of what? Synthesis of protein. Because protein is required, or we can say protein requires two things. One is ribosomes and one is mRNA. So mRNA, or we can say related to that, the information is present in the mRNA, or we can say messenger RNA. So messenger RNA is having all the information that will translate to form the protein. Okay. So mRNA means it's a messenger RNA. Okay. So messenger, do you know? So messenger, what is the function of messenger? Can anyone? So messenger is nothing but the one which convey the masses, or we can say which transfer the masses, which translate something, one thing into the another thing. That is nothing but it's a messenger. So here it's a polyribosome. So we can say polyribosome is more than one ribosome, more than one ribosome present over where? So present over mRNA. So this whole structure we can call it as a what? This whole structure we can call it as a polyribosomes. And this polyribosome, again, it helps in synthesis of what? Synthesis of protein. So here we can say involved in synthesis of what? Involved in synthesis of proteins. Okay, so these are the few things that we can say. And one more thing, what type of ribosomes are there? So here we can say it's a 70 S type of ribosome, and here it's a S is nothing but it's nothing but the sedimentation coefficient. It's nothing but the sedimentation coefficient, or we can say it's nothing but the Swedberg unit. So these are the few things that we can attach, or we can say we relate with the polyribosomes. After that, if you are moving to the next part, so next part we can say that it is also having the vacuoles. The bacteria is also having the vacuoles, but what type of vacuoles? It is nothing but the gas vacuoles are there. It is having what? That is a gas vacuoles. So another structure is nothing but the, we can say it's a gas. So the gas vacuoles which is present here, it's a membraneless or membrane bound. So my answer is, so what's my answer? Do you know? So my answer is, it's a membraneless structure. It's a membraneless structure. While if you are going to the higher organisms, so the vacuole is properly surrounded by some kind of boundary, But here there is no such kind of boundary is there. So here we can say it's a membraneless structure. Okay. So membraneless structure. After that, it is present only in few bacteria. So we can say it's a blue-green algae. You can find this into the blue-green algae, or we can say it's a photosynthetic bacteria. So it is not present in all bacteria, present in few bacteria, or we can say present in some bacteria, not all. So just keep in mind. So this is the gas vacuoles. After that, we can go further. I think in the last class also few students were asking about what? Asking about the inclusion body. So it's nothing but the inclusion body. Because let's say I don't think it's a buffering or not. Like a few students are saying video is buffering. Other is saying no, it's not buffering. So according to Dragon, it's not buffering. While Chaitanya, it's a buffering. Fiona, it's not buffering. So I don't know whether it's a buffering or not, but I think by seeing this video, it's not buffering. Okay. So anyway, it's an inclusion body. Inclusion body means what? Let's say it's a bacteria. It's a bacteria. And if you are a scientist, so after resembling all those structures like flagella, or we can say it's a chromatophores, mesosomes, and many other structures are there. So after that, what will happen? The scientist is feeling tired. Scientist is feeling tired of studying all those structures. So let's say if the scientist is so lazy, or we can say so lethargic. So in that case, scientists will say that chalo theek hai, whatever the structures we have studied is fine, we will publish as such. But rest of the structure, or we can say small structure, we can put into one group. Okay, so the same thing he has done. So inclusion body means what? Small, minute, that is a chinna type of molecules. He considered this as a inclusion body. Okay. So tonoplast, Hatmika, tonoplast. So tonoplast is present in the plant cells, or we can say in plant cell, large vacuoles are there. And this vacuole is surrounded by what? Surrounded by membrane, and that membrane is known as tonoplast. Is that clear? Okay. So next one is, it's an inclusion body. So inclusion body means what? The chemical substance, or we can say the chemical substance, and this chemical substance we can say it's mostly inorganic. So the chemical substance present where? So the chemical substance present in the cytoplasm is considered to be as what? Considered to be as a inclusion body. So this inclusion body, it requires what? Sometimes we can say it's like excretory substance. Excretory substance means what? The substance which bacteria wants to release, or we can say wants to throw out its body. So that is excretory matter. Sometimes it's a secretory substance. Secretory substance means what? Something which bacteria wants to release again. But in this case, it's a very small amount, because secretion is always and always in a small amount. Okay, so that is a secretory substance. Excretory substance, mostly we can say it's a waste materials. So waste materials is nothing but a excretory substance. At the same time, it is having the stored food. Like you can find the bacteria is having what? Stored food in its body, or we can say in its cytoplasm. So these are the few things that you can find in the cytoplasm of bacteria. Okay, so here we can say, so one more question I'm asking, what's a stored food? If the question is coming, so whoever is chatting, or we can say whoever is present online in this class, is having the stored food, but in which form? What is the name of chemical substance that your body is storing as a food? As a food. One answer I'm getting, it's a glucose. No, obviously incorrect. Neha. Chaitanya, starch, again it's incorrect. Glycogen, yeah, Sai Dikshita, yes, it's correct. It's a mixed answer, but the important, or we can say it's a correct answer is, correct answer goes to what? It's a glycogen. It's a glycogen. So most of the heterotrophic organism will store its food in the form of what? In the form of glycogen. But if you are talking about the plants, so the plants is having the starch. So other organisms is also having the different type of stored food. So here I am just writing few names that comes under the inclusion body. So what are the foods? So here we can say it's a glycogen are present in the granules form. So it's a glycogen granules. Thereafter we can say it's a cyanophycean starch. So cyanophycean starch means what? It's a blue-green algae. So whatever the starch, or we can say carbohydrate, the blue-green algae is having, is known as what? Cyanophycean starch, or we can say it's a cyanophycean granules. Here we can say it's a cyanophycean granules. So other than this, so these are nothing but the, somehow we can say it's organic substance. But other than this, we can also add few names. So what are the names? So what are the names? Other name is, here we can say it's a phosphate granules. Phosphate granules. And actually this phosphate granule is what? So phosphate granule is having a lot more amount of energy. So let's say this is the one phosphate, and when this one phosphate attached to the other phosphate, one phosphate attached to the other phosphate, with the help of what? With the help of phosphate bond. And whenever you break this phosphate bond, whenever you are breaking this phosphate bond, what will happen? Huge amount of energy will be released. So same thing is happening with the ATP. So ATP means it is having how many phosphate? So how many phosphate is there? So total 3 phosphate is there. And all the 3 phosphates are, or we can say phosphate is joined with the help of what? With the help of phosphate bond. And phosphate bond is having some kind of energy. is having some kind of energy. So whenever you need energy, just break it. Whenever you need energy, just break it and you will be provided few amount of energy. So again you need energy, so you can get it from the ATP. But again, firstly you have to synthesize ATP. So how to synthesize ATP? So you need to eat a lot more food that I think you are doing every day. So it's nothing but breakfast, lunch, snacks and dinner. So 4 times you are having. So 4 is we can say somehow minimal amount. So here it's nothing but the inclusion body. So likewise we can say these are the small point, or we can say important points related to the prokaryotes. Okay. And next one is, one more thing is Archaebacteria. So do you know something about the Archaebacteria? So Archaebacteria also comes under the prokaryotes. Same as it is, Mycoplasma also comes under the prokaryotes. So Archaebacteria is the one who will survive under an aerobic condition. Under anaerobic condition means where the oxygen is absent, or we can say where the air is absent. Okay, so that is known as Archaebacteria. One more is there, that is nothing but we can say Mycoplasma. So Mycoplasma, we can say it's nothing but the joker of microbiology. So joker of microbiology. So why it is joker, or we can say because it can change its shape. It can change its shape because it is lack of cell membrane. It is lack of cell membrane. So if anyone who is not having the cell membrane, so somehow that structure can modify easily. So here we can say these are the few things that we can relate to this part. Okay. Now we'll move to the next. So I'm just adding few points related to what? Related to Mycoplasma. Obviously Mycoplasma we don't need to add here, but still I'm adding Mycoplasma. It's exceptional part. It's an exceptional part. Why? Because Mycoplasma is saying, see, I don't belong to bacteria, or we can say I don't want to be with the bacteria. So bacteria is asking why. So he said that, or we can say the Mycoplasma is saying I don't have any kind of cell wall, but you people is having the cell wall. So you are somehow stronger and I'm just somehow weak as compared to you. So that's why I don't want to be with you, because you are strong and I am weak. So Mycoplasma is the one which is lack of cell wall. Okay, but it is not having the cell wall. It is not having the cell wall. So what is the advantage of not having the cell wall? So let's say this is nothing but a small pore like a structure, and whenever this one bacteria is entering, or we can say just trying to enter into it. So somehow bacteria is stuck here. But at the same time, let's say it's a Mycoplasma. So Mycoplasma will say, just it's okay, I can enter into it. So according to the pore, the Mycoplasma can modify themselves. So this is the advantage. And what is disadvantage? Let's say you are going, or we can say you are passing on the way, or we can say you are moving on the road. And unfortunately one Mycoplasma comes under your feet. What will happen? The Mycoplasma will burst. But bacteria can somehow tolerate. Okay. So obviously there is a pros and cons of everything is there. So it's a lack of cell wall. So here we can say it can modify itself. That's why we can call it as a joker of this microbiology. Next one is, I'm just little bit introducing, not whole part. So it's nothing but the Archaebacteria. Archaebacteria. So what are the difference between the main bacteria, or we can say Eubacteria, and Archaebacteria? So Archaebacteria, if you are considering Archaebacteria, if you are considering, so Archaebacteria, do they have cell wall? Yes, answer is it is having the cell wall. It is having the cell wall. Again, it's a buffering. I don't know it's buffering or not. So it's not buffering because I can watch this. Anyway, Archaebacteria, it is the oldest one. It is the oldest one, or we can say it's nothing but the grandparent. So one is Eubacteria. So Archaebacteria is what? What is the relation between the Eubacteria and Archaebacteria? So Archaebacteria is the grandparent of what? The Eubacteria. So who comes first? So somehow we can say Archaebacteria comes first. Here we can say Archaebacteria, everything is there, but a few more things. So mostly anaerobes. So mostly anaerobes. And if you are talking about the types, so types is nothing but the. So types is, one is halophiles. Halo means what? Halo means it's an acid. Or we can say it's a. So one we can say it's a halophiles. Halophiles, this is the first one. Next one is it's a methanogens. So methanogens means what? Any Archaebacteria which can convert the carbon, or we can say some other related compounds, into what? Into the methane. So that is known as methanogens. That is known as methanogens. Have you visited the beach area? Have you visited the beach area? So there you can find some marshy places. There you can find the marshy places, or we can say some mangrove area is there. Okay. So the mangrove area is good or bad? So mangrove places is good or bad? So for us, it's not so good. Why? Because you can find a lot more Archaebacteria, or we can say it's a lot more, we can say methanogens there is there. So whenever you go to that place, it produces methane, or we can say methane will somehow pollute the air. Pollute the air. So that's why means we should avoid this kind of places. We should avoid this kind of places. So for us it's not good, but for other animals, or we can say for other plants, or for other species, it's good. Okay. So here it's one is halophiles. Another one is methanogens. Okay, and methanogens, one more places where you can find the methanogens. So methanogens is also present in the gut of the cattle. Cattle, or we can say it's a buffalo, or we can say it's a cow. So few animals, or we can say few animals, whoever is eating cellulose. So cellulose is not digested by that animal. But this methanogens can digest the cellulose. So methanogens is good friend of cattle, or it's bad? Methanogen is good friend of cattle, or it's bad? So I think it's a good friend, because cattle is having a lot more grasses, and grass is having what? Cellulosic component. And that cellulosic component cannot be digested by what? Cattle. Cannot be digested by the cattle. So methanogen saying to the cattle, okay, it's fine, I will digest the we can say cellulosic part, but just give me some spaces. Give me some spaces to live inside your body. So you just give me food, I will digest, and somehow you'll convert this into the energy. So obviously both are getting benefited. Both are getting benefited. One more is there, that is known as thermoacidophiles. Thermo means what? It's related to temperature. It's related to temperatures. And acido means that is acidic, means that is a pH. So the one who loves to grow, where? Who loves to grow in high temperature as well as high acidity, or we can say it's nothing but it's acidic condition. So this is known as thermoacidophiles. And we can say it's like a sulfurous spring, or we can say hot sulfurous springs are there. So there you can find this type of organisms. So likewise, it's nothing but the introductory part of what? Prokaryotes. Now we'll start with the introduction of the eukaryotes. And eukaryotes is nothing but the we are talking about ourselves, because we are nothing but we consist of what, or we can say what are the type of cells present in our body. So what are the type of cells present in our body? So we can say it's a eukaryotic cells. So firstly, we have introduced something about the prokaryotic cells. Now we'll discuss something about the eukaryotic cells. Okay. So firstly, let me differentiate what is prokaryotic cells and what is eukaryotic cells. So once we will start with I think it's a U. U means what? It's a true. Dragon, no, it's a botany, right? And anyway, like this cell biology, it comes under botany here, but in real meaning, it is useful for both botany as well as zoology, or we can say it's nothing but it's a part of biology. It's a part of biology. So here it's a U means what? It's a true. And karyo means what? Karyo means what? It's a nucleus. So anyone who is having the well-defined, well-organized, advanced type of nucleus, that is known as what? That is known as eukaryotes. That is known as eukaryotes. And can you tell me what are the organisms included here in the eukaryotes? So obviously you can name the kingdoms. So directly I am writing the kingdom. So eukaryotes includes what? Includes, I think, 4 kingdom. According to you, it's a 4 kingdom. So what are the 4 kingdom? Tell me. Unicellular eukaryote is known as what? Or we can say we can place into which kingdom? We can place into, yes. Atmakuri, yes, it's a protista. Chalo, theek hai. Unicellular eukaryote placed into the protista. Next one is, after that, any other name? Fungus, it's a prokaryotes or eukaryotes? Fungus is prokaryotes or eukaryotes? Fungus is eukaryotes. Okay, good. Next one is animals, or we can say we. Prokaryotes or eukaryotes? Again, it's a eukaryotes, and plant is also eukaryotes. So what are the things we can include into this part? So the first is the simplest eukaryote is present, or we can say placed under which group? That is kingdom protista. Kingdom protista. After that, little bit advanced, or we can say little bit complicated as compared to the protista is fungus. More complexity will be there. More complexity will be there. So it is more complex. It is considered to be as a plantae. It's a kingdom plantae. After that, what's there? The last one is nothing but the Animalia. If the question is coming, how many of them are multicellular? How many of them are multicellular? 1, 2, 3, or 4 here. How many of them are multicellular? So what's your answer? 1, 2, 3, or 4? I'm just getting mixed answer, but Kamakshi, it's your answer is true. Neha, it's also true. Okay, Lakshmi, also true. So answer is 3. So why? Because fungus, plantae, animalia. So fungus, plantae and animalia are only 3 multicellular. While if you are talking about the protista, you only said now because protista is unicellular, no doubt it's a eukaryotes, but it's a unicellular. So here these are the few parts. Now we'll move to the next one. Next one, what are the characteristics of eukaryotes? So the features, or we can say in other word, characteristics of eukaryotes. So the first characteristic we can add is, can you tell me like the main characteristic, or we can say the main thing which is known for? Because it is popular for what? I think it's a popular for the true nucleus. That's correct answer here. It's nothing but the nuclear membrane. So here we can say nuclear membranes are present. Nuclear membrane is present. Now what's the next? Can we say that one word we are using? So what's that word? Can we say that the whole cytoplasm is divided into the small compartment, or we can say small rooms? So let's say one room we are giving to the mitochondria, other room we are giving to the we can say chloroplast, other room is giving to the vacuoles. So here we can say, what's that? The whole cytoplasm, or we can say the whole space, is divided into the small compartment. So this word we can call it, or this process we can call it as a compartmentalization. So compartmentalization means what? That is a dividing into the compartment, or dividing into the boundaries. So here we can say it's a compartmentalization are present, or in other word we can say membrane bound organelles are present. Membrane bound organelles are present. What else? Do they have true chromosome? My answer is yes, it is having true chromosome, because true chromosome requires what? One histone protein, means proper packaging is required. So whenever you are packaging, whenever you are folding the DNA, so what type of protein is required? I think we have discussed in 1 or 2 classes. The protein required to fold your DNA, or we can say it's a packaging of DNA. Yes, Atmakuri, packaging of DNA. Yes, Neha, it's correct answer, histone protein. Kavya, it's a histone protein. Correct, Datta. Yeah, yes, it's a histone protein. So histone protein is the one which helps in folding of what? Which helps in folding of the DNA. Because can we say that DNA is this much huge? DNA is this much huge. In this case, what will happen? So if DNA is approx, we can say DNA is 2.2 meter. Yes, Neha, it's your correct answer. It's a 2.2 meter of DNA is there. And this 2.2 meter of DNA is distributed among how many chromosomes? So if we are human beings, so 46 chromosomes are there. So if you just collect all 46 chromosomes and just try to pick out the DNA, and after adding the length of all the DNA, you can find it's a 2.2 meter. So 2.2 meter of DNA, if you want to store in very small places, you need to do what? You need to properly pack it. And for that, which helps? That is, a histone protein is helping. Okay. So is that clear? So here we can say histone protein is present, or we can say true chromosomes you can find. So it's a true chromosome, or here we can say it's nothing but the histone protein is present. histone protein is present. After that, we'll move to the next part. So what's the next part? Because whenever you are going for the complexity, you need some kind of support. Either it's a structural support, mechanical support, or many other supports is required. Because if you are moving to the complexity, complex thing requires many supports. So what supports? So here we can say it's a cytoskeletal structures are there. So cytoskeleton structures are present. Okay. So after the cytoskeleton structure, next one is, do they divide or not? So we can say it will divide, but the type of division, because in prokaryotes, it was dividing by what? It was dividing by binary fission. Means 1 turns into 2, 2 to 4, 4 to 8, means every time the things becomes double. But in this case, what will happen? We are using some kind of a special thing. So what's that special thing? So a special type of division is there. One is mitosis. So mitosis and meiosis both are present. So what is the uses of mitosis? Can we say that mitosis, it helps to grow? So can we say that everyone, what is the use of them? So which one? Versika, you are asking some question, what is the use of them? So what's them? It's a cytoskeletal structure or something else? So here we can say one is mitosis and one is meiosis. So mitosis, what is the uses of mitosis? Main uses of mitosis is to grow. So can we say that everyone started their life with a single cell? Can we say that we all have started our life, or we can say started our journey from single cell or multi-cells? So can we say it's a single cell? And that single cell is nothing but a zygote. Because whenever we are saying it's a life, so a single cell can also have the life. A single cell can also have the yes. Cytoskeleton means what? Cyto means what? Cyto means it's a cell. Skeleton means what? It is providing some kind of rigidity. It is providing some kind of skeletal. Like I am having the bone as a skeletal. I am having bone as a skeletal system. Same as is, cell also requires some other things. Cell also requires, or we can say, to maintain the structure, to maintain the shape, it requires what? It requires some kind of support, or we can say it's a mechanical support. Is that clear? Next one is a meiosis. So in the case of meiosis, what will happen? Like how it is helping? So meiosis, it will help to reduce the number of chromosome. Or we can say it helps in the formation of gamete. Or in other words, we can say it helps in sexual reproduction. So mitosis and meiosis both are having its own importance in their life. So after that, we'll move to the next. Again, it's a Flagella, that is a locomotory structure. If it is locomotion, then locomotory structure is nothing but the flagella. Sometime you can find the cilia, and flagella is nothing but the, here just I am writing 9 plus 2. For now I am writing 9 plus 2. So this 9 plus 2 is the arrangement of microtubules. Arrangement of microtubules is nothing but the 9 plus 2. So I am not explaining, or we can say I am not going into details. 9 plus 2. So 9 plus 2 is nothing but the, it's a microtubules, or we can say it's an arrangement of microtubules. Okay. Meiosis, Alavina, Lakshmi, like meiosis, the main function of meiosis is to reduce the number of chromosome. Like if anyone is having chromosome number is 2. If anyone is having 2 number of chromosome, then if it is going for the what? Going for the meiosis, the number becomes 1. Or we can say if you are having 46 chromosome, then after having this meiosis, it turns into what? It turns into the 23 chromosome. Next one is, what is mitosis? Mitosis is the division which helps in growing, or we can say wherever you are growing, or we can say wherever you find the growth, it is due to the mitosis. And meiosis is it is for the reproduction, or we can say making the chromosome number half. Okay. So here these are the parts. So here it's a 9 plus 2 I have written. 9 plus 2 is nothing but the, its arrangement. 9 plus 2, it is nothing but the arrangement of what? Arrangement of microtubules. Arrangement of microtubules. So arrangement of microtubules is nothing but the, here you can find the flagella. Flagella arrangement is 9 plus 2. So 9 is somehow we can say it's a doublet, means 2 microtubules will be of 9 and 2 is of single. So can we say that's a 9 into 2 is 18? 9 into 2 is 18 plus 2 into 1, again it's a 2. So total microtubules how many? Can you count? Can you calculate the number of microtubules? Like 9 doublets means 9 into 2 is 18. 2 into 1, again it's a 2. So 18 plus 2, that is a 20. 18 plus 2, that is a 20. Yes, correct answer, 20. So after that, we'll move to the next. And next we'll start directly with the plasma membrane. That is the most important structure. The most important structure is plasma membrane. And first time who has talked regarding plasma membrane? I think in the first or second class we were talking about this. First or second class we were talking about the plasma membrane. So who were the first scientists talking about the plasma membrane? So can we say it's one is zoologist, and the name of zoologist is Swan. Theodore Swan was the first one who were talking about what? Who were talking about the plasma membrane. Okay, so just let's go with the plasma membrane. And if you are talking about the plasma membrane, so it's better to go for the cells. It's better if you really want to study the structure of what? Want to study the structure of plasma membrane. Plasma membrane. What type of cells you will prefer? I am just giving you two options. One cell with more number of organelles. another cell with less number of organelle. So I am giving you two options. Again, I am repeating my question. You want to study the structure of plasma membrane. You want to study the structure of plasma membrane. You are having two options. Either go with the cells with more number of cell organelle, or the cells with less number of cell organelle, which is more preferable for you. Yes, that's correct. Neha, less cell organelles, or we can say the cells with less cell organelle is convenient for you to study the plasma membrane, or we can say structure of plasma membrane. So in your body, I think RBC is the best example. RBC. So it's better to experiment on what? Experiment on RBC, because RBC is having very less number of organelle. RBC is having very less number of organelle. One more is Rama Devi. Why not more organelles? If you are going for the more organelles, then obviously this whole process becomes complicated. You want to study the plasma membrane, but along with the plasma membrane, other organelles will also come. So that's why it is somehow becomes complicated for you to study. So is that clear to you? Why we are not taking the cells with more organelles? Why we are taking cells with the less organelles? Okay. So now we'll move to the next one is plasma membrane. So the features of plasma membrane, if you are talking about the features of plasma membrane, first feature is it's selectively permeable. So why it is selectively permeable, and what is selectively permeable? So selectively permeable means what? It allows only the things that the cell requires. It allows only the things that cell requires. Okay. So that is nothing but selectively permeable. Selectively permeable. Okay. After that, it's a dead or living. So here we can say it's nothing but it's a living part. Why it is living? Because metabolism is present. That's why it's a living. Next one is it's a thin and delicate. Obviously, it's a vulnerable. Once you can break it easily. So here we can say it's a thin and delicate structure. If you are talking about what? If you are talking about the plants, so in that case, cell membrane is surrounded by cell wall. Cell membrane is surrounded by the cell wall. But if it is animal cells, it is directly exposed to the surrounding. Means in that case, the cell membrane is not surrounded by the cell wall. Why? Because animal cell is lack of cell wall. While plant cell, the cell membrane is surrounded by what? Cell membrane is surrounded by the cell wall. In the same way, fungus is also like cell wall is present. So in fungus also we can say cell membrane is surrounded by what? Cell wall. So can we say that plants and fungus, in both what will happen? It is surrounded by cell wall. While in the case of animals, it is not surrounded by the cell wall. Next one is CSK. What is protist? Protist is unicellular eukaryote. And obviously it's a mixed features are there. Okay. So next one is if you are going to the compositions. So compositions, it consists of what? The most important is protein. So protein is present in huge amount. Protein is, or we can say maximum percentage is protein. Maximum. After that, what is, so lesser than this is lipid. Lesser than this is lipid. After that, some carbohydrate is also present. Some carbohydrate is also present. So these are the few compositions. So maximum is protein, lesser than is lipid, lesser than is nothing but the cell membrane is present in animal cells. Yeah, cell membrane is present in animal cell, or we can say cell membrane is present in all living beings. Okay, so that's why you can make one more point, or you can say you can add one more point. Cell membrane is what? It's a universal, or it is present in only few organisms. So again we can say it's a universal. It's a universal. So after that, if you started studying, that is what? Class 11th. In the class 11th, they will briefly introduce. It will briefly introduce, like who has given, like lot more scientists come forward and just started explaining their views. Started explaining their views. So obviously, some scientists having logical views, or we can say the views were accepted. So for some scientists, views were it's popular, it's accepted, while others' views were rejected. Why? Because the scientist was not able to explain everything. Not able to explain everything. So that's why here just I am writing one of the most popular, and actually this question is too much, we can say it's a popular in NEET. So directly they are saying one question, most popular model to explain what? To explain the structure of cell membrane. Structure of cell membrane. So if you answer correctly, then how many marks will you get? So you will get 4 marks. You will get 4 marks out of 720. So what's the answer? So the answer is fluid mosaic model. So fluid mosaic model was the model that was presented by, so the model name is fluid mosaic. So it's nothing but the fluid mosaic model. Yes, Sadhna, that's correct. Singer and Nicholson was the two scientists who proposed this model. So Singer and Nicholson are the two scientists who proposed this model, or we can say who just explain about the cell membrane. Here directly I can write proposed by what? Proposed by Singer and Nicholson. So Singer and Nicholson was the scientist who has given this type of model. So anyway, like we'll stop the class, today's class. We'll stop today's class, and in the next class we'll start with what? Start with the structure of cell membrane. And anyway, in the cell bio, the structure of membrane is most important. Structure of the cell membrane is most important. That we will discuss in the next class. Okay. So till now, bye bye everyone. We'll meet you in the next class. Take care.